Good afternoon, and thank you for joining us for the 8th episode of our ESHG webinar series. I'm Bill Newman. I'm the current Vice President of the ESHG, and it's my absolute delight to introduce our speaker today. Our speaker today is Professor Lisenka Vissers, who's Professor of Translational Genomics at the Radboud University in Nijmegen in the Netherlands. Lisenka studied biology in Nijmegen, and then she went on to get her PhD when she was doing research on microarrays, which were something that we used lots more in the past, but have been completely transformative over the last 2 decades in the diagnosis of the causes of developmental disorders. But Lisenka, being Lisenka, has very much moved with the times, and first using exome sequencing and more recently genome sequencing to unravel the causes of rare developmental disorders. And particularly today, talking about one of the latest developments is a really exciting about the opportunities of using long-read genome sequencing. So Lisenka, thank you very much for joining us. If you have questions for Lisenka, there will be a little box on the bottom of your screen for the question and answers. So please put any questions in there rather than in the chat, and we'll open that up a little bit later during Lisenka's talk. So all it leaves me to do is to say thank you very much, Lisenka, for joining us and presenting today.
Well, thank you so much, Bill, for this very nice introduction. And in the 2 minutes before we started this meeting, I said, well, I think we still know how to share our screens, but I'm going to see whether I can actually still get that done. That you not see my presenter mode, but you actually see my screen for presentation.
Yeah, that's perfect, Lisenka.
Good. So today I'm going to talk about the opportunity of long-read sequencing and to use this in a clinical diagnostic setting. And first of all, I would really like to thank the European Society of Human Genetics of giving me the opportunity to present our exciting work here to you today. First of all, I would like to mention to you that many of the data that I'm going to show you today are part of a joint collaboration between PacBio and Radboud UMC. And with that, PacBio has kindly contributed to this project by supporting us with reagents and also some in-kind contributions and expertise. And by no means, however, the data that I'm going to show you today are their opinions, but ours, and they have also not been involved in any of the conclusions that we have been drawing from our data presented. So with that, I'd like to start with the introduction. And as I'm probably preaching to the wrong choir, you are all aware that there's at least 7,000, if not 8,000 rare diseases, for which many of them are of genetic origin, manifesting throughout the early childhood ages. 80% is expected to be genetic. It's a lengthy process to find a diagnosis, if found at all. So approximately 1 in 3 patients only gets a diagnosis today. 30% of children still die from the disorders. The diagnostic quest is still very long, and sometimes may even take up to 6 years. Many different medical specialties are involved. Some of the patients see more than 10, but the main question remains, what is the cause and how can we find it best? If we look back, and this is also where Bill alluded to in the introduction, on what next generation sequencing has offered to us, we can clearly distinguish a genetic diagnostic yield, if you want to say, for rare diseases in the pre-genetic next generation sequencing era and the post-genetic diagnostic testing using next generation sequencing. So the data that I'm showing you here is of a cohort that was followed in time and in parallel to standard of care exome sequencing was performed. And in the same type of patients, doing the exact same things at the exact same moment, doing simultaneous to standard of care, the exome analysis, 1 in 3 patients got a diagnosis, whereas in the pre-NGS era, only 7% got one. We have put lots of efforts in what is the best next generation sequencing test. Is it exome sequencing or genome sequencing? And in order to at least look at it from our perspective in our diagnostic settings, we therefore also looked at similar comparisons of looking at exome sequencing simultaneously to short-read genome sequencing. So again, a cohort of 150 patients followed in time using similar type of technologies in exome sequencing, and in parallel, the short-read genome sequencing technology used. At the time, relatively disappointing. We could only see in the same group of patients a 2% increase in diagnostic yield. But nonetheless, we said genomes are very likely the future, because if we look in time, irrespective of what next generation sequencing technology we use, and we follow the same patients again, 5 years after the first initial analysis, where 1 in 3 patients get a diagnosis, you can add approximately 20% by systematically reanalyzing the same cohort, by using new tools on the data that was already generated, but also, of course, the updated interpretation of novel disease genes, new information that has become available on variants that had already been identified, leaving still a large majority unexplained, but also showing the value of novel technology and increasing knowledge over time to look at our patients' genomes. Knowing that knowledge is the main driver for this group of individuals now getting a diagnosis over time, we said it is actually very useful if we start using genomes, because if we have genomes, the wealth of information and availability of data that becomes available to apply this knowledge to is the best that is out there. So a couple of years ago, we set ourselves in Nijmegen the goal to see whether genomes are the most optimal diagnostic test to be using for rare disease patients. And by that, optimize the efficiency in our diagnostic setting, but also increase the diagnostic yield. So with these two goals, we set ourselves a study where we looked at all of our patients with the standard of care technologies here highlighted by different icons, and to see whether we could then, by replacing all of this, using an exome or a genome, whether we could actually increase the diagnostic yield. At the time, we were not entirely sure what that increase should look like or what was to be expected, and whether we would actually surpass the 2% that I just showed you on the previous slide. But this is what the goal was at that time. If we look at the data, we took a whole series of patients from the year 2022 and analyzed them in all of the details that we could. This included over 800 different clinical indications for which patients were referred to our hospitals for diagnostic testing. And in total, we tested nearly 25,000 patients that were seeking clinical diagnostic testing for their rare genetic disease, assumed to be of genetic origin in the germline. If we look at this cohort, we unfortunately only saw an increase of 3 to 5%, which was very marginal, but still relatively feasible. But unfortunately, we could not entirely see that all of our diagnostic patients could be moved to genomes, and this is here highlighted in the bottom below. So of all the 25,000 cases, only 17,000 individuals were actually amenable for a short-read genome technology, simply because the variants that were identifiable in the other 7,000 could not be retrieved in the studies that we did. So unfortunately, this meant that despite our wish to go to a genome-first approach, we found that short-read genomes were not the best way to go. This was almost at the same time that long-read genomes found their first papers on larger scale case studies, where they showed, based on clinical pre-selected cohorts, that an increase of 0 to 20% could be achieved. So this is when we said perhaps we should not just go for short-read genome sequencing as the test to go, but perhaps we should evaluate whether long-read genomes are actually better as a transfer to go to, also making us more future sustainable for the reanalysis, increasing knowledge. At the time, there were two main players, and I think that's still the case. If you would go for long-read genome sequencing, and I will not go into all of the details today of both technologies, but just to let you know that there's multiple ways to look at the long-read genome, and what essentially is the difference. So we have the PacBio version, which is based on hi-fi sequencing, and we have the Oxford Nanopore sequencing technology of ONT. Irrespective of the technology, the main thing is that it's a single molecule-based technology. You can do real-time sequencing, and the reads are far beyond any size of what you can generate with a short-read genome equipment. Because of that, and I'll show you some more details later, the fact that you can now generate the longer reads allow you to give a better overview of the human genome all at once. And that is therefore also expected to be one of the biggest pushes for the rare disease field since exome sequencing was first launched in the the end of 2010, early 2010, 2012. So if we look at the basic principle and how it is different from for instance, short-read genome sequencing, of course, in short-read genome sequencing, your read length is approximately 300 base pairs. If you do 2 times 150 paired-end sequencing reads, you can generate the mapping to a reference human genome. You can put some pieces of the puzzle together, but the pieces are still relatively small, and it's very difficult to see the entire puzzle as a whole. Whereas if you look at the long-read genome technologies, here depicting the read length for the PacBio hi-fi sequencing, you can generate read lengths of approximately 20 kb. And this means that the puzzle gets easier to be made. There's little to no more gaps in the genome. You have a contingency. It allows you to go for phasing and haplotyping instead of just coverage-based variant calling, allowing you to have a better representation also for the repeats and the homologies that are also present in the human genome, and especially a variant type, which was very difficult to detect from the short-read technologies, collectively termed the structural variants and the repeat expansions, are of course much easier if your read length exceeds 300 base pairs. The bonus of long-read sequencing for both ONT and PacBio is, of course, that there is a DNA modification that you can also get as a readout, which is the methylation 5-MC. So with that more or less covered, the question became relevant in our setting, and that was whether this would be the ultimate test that we were looking for to replace all of our current diagnostic testing. And then we had 4 or 3 different projects to see whether this is the way to go, and I'm going to present to you the data of these studies here today. So the clinical studies to test whether hi-fi genomes, here abbreviated throughout the rest of my presentation as lrGS, is better or worse, of course, than any of the other technologies that we have available, is that we have 3 different cohorts. The first cohort is a series of 100 samples where we know that short-read genome technologies are challenging to find the relevant variants to see whether then long reads do better. We have cohorts of 200 patients together, 100 trios and 100 index or singletons or larger families, who have reached the end of their routine care to see whether long-read genome sequencing is able to increase the diagnostic yield. And if that is all positive, we set out to do, in parallel to standard of care, 1,000 representative diagnostic samples of a year 2022 again, in parallel to routine standard of care testing, to see whether we can then predict what the outcome ultimately is of long-read genome sequencing, should this be used as a first-tier test and only test in a generic first-tier long-read genome sequencing diagnostic approach. So let's start with the question whether long-read genome sequencing handles the clinically relevant variants in more difficult to sequence regions by short reads in the genome, and whether they do better. To give you a flavor of what we then did is that from the study that I just presented to you in the introduction part, where we said, well, genomes are great, but we cannot replace all of our diagnostic testing because we cannot find all of the relevant clinical variants. We actually expanded on that variant sample. And instead of sequencing just a few, we took 100 individuals together holding 145 different variants in what we at that moment considered to be one of the most difficult regions of the human genome to sequence for various reasons. So one of the reasons was that there is there's a couple of loci in the human genome that are of importance for rare diseases, but are dealing with homologous regions, so the pseudogenes. And of course, in short-read technology, it becomes very difficult to then map those short reads to the reference genome and determine where the variant is located, whether you're looking at the pseudogene or whether you're looking at the functional one. So we selected 16 different loci that are of importance to our diagnostic series of testing, but probably for many other diagnostics labs too. They are listed here on the left-hand side. There's an additional series of cases or genes that are very difficult to sequence using short reads because they deal with repeat expansions, generally exceeding the read length of a short read technology. And these are listed here as well. So we had 14 different genes together being tested for 25 different index cases, holding 26 variants, because some, of course, may be compound heterozygous, and so on and so on. So we had 23 different complex rearrangements consisting of translocations, etc. We have imprinting loci because we wanted to test whether the 5-methyl-C readout was possible, etc. And important to know that if you do this, at least in our diagnostic lab, to find the relevant variants in these cases, we use on average 1.5 or so different genetic workflows, so tailored to find the different variants of relevance. Having said that, we just sequence them out using one SMRT cell per sample, aiming at the coverage of a 30x long-read genome. We use the standardized callers that were then provided by PacBio. So we use deep variant for the SNV and indels, the hi-fi CNV for the CNV calling, paraphase for those regions of containing pseudogenes and homologous regions, and the SVs were called by PBSV, and target is a special dedicated tool that I'll show you in a minute for the tandem repeat expansions. If we look at our first experiences on how sequencing went, we actually noticed that we did not have an awful lot of resequencing to be done, so it was quite robust to sequence those 100 samples on a single SMRT cell each. We had an average output of 94 gigs, and on average, we indeed reached 29-fold of coverage, aiming for the 30 per SMRT cell. If we look at the read size, we see a very nice histogram or distribution with the average read size of approximately 15 kb, which was for the SMRT cells used and the chemistry used at the time, what we were expecting to reach. And what I'm going to show you now are just a few examples of what then the data look like, and to get you some more familiarized with long-read genome sequencing. So what you see here is a screenshot from IGV, and IGV is a tool where you can show your reads after sequencing. You see red ones and you see blue ones, and the red ones and blue ones are visualizing the different haplotypes, which is one of the main advantages of looking at long-read genomes. And what you can see here is that there's something going on, and boxed here in the red square. And this is a case that was sequenced because of a patient having a myotonic dystrophy in the routine report in standard of care being tested by dedicated targeted assays. And this is caused in this patient by a repeat expansion in the gene CNBP. And in routine care, you then say the repeat length is longer than 75 repeats. But now having the access to the long-read genome data, you can see not only that on which allele this is located, and this is very nicely shown. But if we zoom into what is happening here, you can also see that the number is represented on how many repeat copies is presented. And if we use this tool called target that I already alluded on in the previous slide, you not only get the exact read copies, so there's a repeat copy of 317, but you also get the two alleles visualized by which repeat is causing this repeat elongation. And this is then visualized by repeat context. So you can here see that the repeat extension is caused by the CAGG repeat, as expected in this case. There are no interruptions, and you can get the individual molecules here shown by the reads. So this is much more information that you would get in routine care, where you now not only have the exact number, which is only stated in routine care by more than 75, but also the exact sequence context, verifying that this is indeed not just a repeat, but also the repeat that is clinically of relevance. Looking at the imprinting and methylation, you can see some similar kind of nice experiences that you get from looking at long reads, again, visualizing haplotypes. What you see on the left-hand side is an example for imprinting disorder. So this is the maternal UPD14 causing the problem with the MEC3 gene based on methylation of the parental sample of one of the maternal and paternal alleles. So what you would normally see is that you have here the MEC3 genes, and we're here now colored and grouped by haplotype. So again, which reads from which chromosome they are originating from. And the colors here represent whether the CpG that was highlighted is either methylated or non-methylated. And what you would see now is that both of them are methylated, which is in this case causing the problem in this index case. If you can do this for imprinting disorders, you can also look at the X chromosome inactivation. And what we had in our series were a couple of samples where the gene was, or the mutated variant, or the mutated gene, or the causal variant was located on the X chromosome. So you would expect in female samples that this is leading to a X inactivation skewing, because you preferably do not want that gene to, or that chromosome to be active. And what you see here is a list of all the 50 or so samples that were of female origin in this series, where more to the right, there is more skewing, and more to the left, there is more random skewing. And what you can see is that the individuals with the X-linked variants are ranking more towards to the right. And interestingly, we had 2 other samples for which we saw that there was also a presumed skewing of the X chromosome, and we could not explain this until we started looking at which samples they belong to. And these are in fact 2 females that were the mothers of sons who had actually an X-linked variant that was pathogenic. And of course, you would expect that in those mothers who passed on that variant, there's also an X-linked skewing going on. So this actually confirms that we could actually see this very well in our data series as well. If we summarize this part on what we could find and what we couldn't find, looking back again at the different reasons why we included the sample in this series, there's a few striking observations. So one of them is that 83%, highlighted in green, are variants that are automatically detected from all of the tools that PacBio at that moment had in their armamentary to just analyze. And there's an additional 10% that we could see in the data, but for which no tools were available at the time for routine random detection. But there's an additional thing that you can notice from this slide, and that there's a couple of variants that we could not detect. And they are not just randomly distributed across these series of type of variants, but they're actually belonging to dedicated classes. So 4 of the variants that we could not find were related to complex rearrangements, and they dealt with Robinsonian translocations and repeat-mediated balanced events. So whereas unbalanced events are not a problem at all for long-read genome sequencing, if a balanced event does not lead to any loss of material, of course, and the repeat which mediates the event is longer than the repeat length of what is being generated by the sequencer, it becomes very difficult to, again, find where the break is occurring. And then, of course, you miss the balanced event. The other thing that we notice is that within the number of repeat variants that we aimed to identify, approximately 20% could not be identified. And those were not randomly distributed across all of the repeat motifs, but all had to do with variants, repeat expansions dealing with GA repeats. And the reason for that is that GAs cause so-called G quadruplex formations. And this, of course, leads to instability of the polymerase when sequencing the read, and then therefore drops off. And that gives you no information of that locus, or less information, so less reads being mapped to that locus. And the longer the repeat, the more likely it is that the polymerase falls off and causes this instability. So it is very difficult to then detect those GA repeat-based expansions. The other thing that we just talked about was the skewed X inactivation, where you noted that some of the samples were not strikingly ranking on top of all the way to the right. And we believe that this is either not the skewed inactivation as expected, or whether perhaps the original standard of care test based on just a single CpG in one of the genes, in the AR receptor gene, may not be the best test to look at this location. But now we have the information of the entire X chromosome, perhaps providing more sensitive information, showing you that they might not actually be skewed. But all in all, what we got here led us to believe that this was actually quite okay. The next question that we wanted to answer is, if you have a 30x genome, long reads, do you need 30x, or can you perhaps also do with a little less, given that the cost of long read sequencing is still it's not at par at short read genome technologies. So if we can do with less reads or less coverage, perhaps we can then also reduce some of the costs still associated with this technology. So for this, we made a model in which the bioinformaticians randomly sampled from all of the cold variants a down titration of 30x to 20x to 15x and 10x to see what the impact would be on the overall detectability. So in this series, we only used 177 variants that were cold. So the GA repeats were not included, because if you cannot call it a 30x, you sure cannot call it a 10x. And for all the variants that you can call, you can see this very nice titration curve going down. So at 10x, you only detect approximately 77% or so of all the variants that you could detect at 30x. Where 30x is 100, going all the way down. So at 20x, you could potentially say that you can still detect approximately 96% of all of the variants that you originally called, whereas it's only 89% if you use 15x. This is for all the variants. So we also looked at, is this any different for the different variant types or the different variant colors? And what you can, of course, see that this is the graph I just showed you a minute ago. But if you look at the different variant types and the different colors used for that, you can see that this is not equally distributed or equally going down in a similar rate for all the different variant types. This is in part due to the complexity of those variants, but also because of the numbers that deviate between the different variants that we have used here in this calling algorithms. Finishing up this part of the story, I think what we at that moment decided is that we can clearly say that if we look at the most difficult regions of the genome where clinically relevant variants may be located, we believe that long-read genome sequencing at 30x is very well able to call those variants. And with that story finished, we said, okay, is this unique to us? No, it's not. If we focus even more on certain variant types, we also collaborated in the Hi-Fi Solves Consortium, where we, across Europe and other parts of EMEA, collectively looked at a sort of similar kind of study where we had 127 different variants in 86 samples, again, for the same kind of reasons included as before, but are now only located to the pseudogenes, and we get similar results. So this is not unique to us, but this is these results are generated on any Revio sequencer at the moment. So also here, what you can see on the left right-hand side is that all the variants can be called apart from one. But if you really look into the regions that have the high homologous regions, that you need a dedicated caller, here called paraphase, to find those 31 variants that you would otherwise not find, but all these tools are readily available at the moment. If we now know that we can detect those regions very well, the question is, can we then also increase diagnostic yield? Because that was one of the other aims of our study, increase the efficiency by looking for one test fits all, but also at the same time seeing whether we can actually increase the diagnostic yield for rare diseases, given that only 1 in 3 to 1 in 2 sometimes only gets a diagnosis. And this requires a different study design to say, because now you're not looking at whether you can replace one of the tests being performed, but you want to make sure that you test a cohort that is still negative after all of the routine diagnostic care that can be performed, whereas there's a strong clinical suspicion of a certain phenotype for which genetic heterogeneity may be very limited, and that you say, well, this patient has this disorder, but I still have not found the relevant variant. So for that, we had such cases where there is a clinical diagnosis very strongly made, either because it's a dominant one where we miss one variant, but sometimes also families where we know that it's a recessive disease where we miss the second variant. And of course, helping us in filtering the data and prioritization of variants. We also looked at what we call the de novo paradigm for neurodevelopmental disorders, where we can limit the search space by only studying de novo variants in those trio-based approaches. If we first look at those 100 trios, we selected 100 trios of neurodevelopmental disorders. The cohort was approximately 50-50 males, females. The age was relatively considered a pediatric cohort. We again did a trio-based sequencing, one sample per smart cell, yielding the same kind of data from a single smart cell, but also resulting in the same type of coverage as before. We have again used the standardized pipelines with our in-house annotation tools, and we focus for this cohort specifically on variants that are causing de novo mutations and analyzing therefore only the low-hanging fruit, if you would like to say. If we look at the numbers first before we put any clinical relevance to the variants, we can see that if you use a long-read genome trio-based approach, that approximately 68 substitutions are identified, 4 indels smaller than 50 base pairs, and approximately 3 structural variants. So the numbers are fully in line with the recent publications in Nature and Nature Methods on how many variants are seen if you have a very high-quality genome. So from this, we would say that we have pretty high-quality genomes to study. So for all of these variants, we subsequently looked into the relevance to their disease. And without going into all of the details, because we literally only looked at the low-hanging fruits, so looking at those variants that are easily detectable in genes for which known genotype-phenotypes correlations exist, or because they are so large or so disruptive to gene function that they must have a relation to disease. We can see that we can approximately increase diagnostic yield between 6 and 16%. So 6% are the structural variants mostly in indels that are really in the known disease genes, whereas the other, you can see again, an overrepresentation of the structural variants that are still possible causes at the moment, because we still need to further study how the relation between these disease genes and the phenotype really occur. I'll show you an example of those. So this is one of the cases which is who has a severe dysmorphic features, hyperterrorism, macrocephaly, a mild moderate intellectual disability, and psychological behavior. And in the data, we see a call that says 4 de novo B and D calls, and this is what you then get as an output. And B and D stands for the break ends, and these represent discontinuity of the reads of the sample alignment, suggesting that there might actually be a translocation or unresolved structural variant going on. And if we zoom into these loci again in IGV showing the reads, you can see the first allele and the second allele. And in this allele, you can see that there's something going on here on chromosome 2. There is coverage generated by reads that by the rainbows do not seem to be located there, because otherwise they would have been looking in a gray color. So there's additional sequence located here on chromosome 2. And if we start looking into these reads on where they were coming from, we can see that they're actually duplicated sequences from chromosome 3, where they have been reversely orientated, inserted into this chromosome 2 locus. So although this is still relatively large, we did not identify this on the short read genome. And if we look into the context of relevance into the patient's phenotype, we know that this gene is not yet associated to human disease phenotypes, but looking at all of the population metrics and gene function, we would say that this is definitely a candidate disease gene for neurodevelopmental disorders. So having one of these examples in our series that had not been identified as of yet, we're very comforting to see that long-read genome sequencing may be able to increase diagnostic yield. Again, we have looked ourselves into the neurodevelopmental disorders, but through other collaborations in SolveRD, which is a large European consortium looking at similar kind of efforts, we can see that if they studied a cohort of 93 cases with different types of phenotypes, including neurological ones, neuromuscular ones, and epilepsies, they obtain a similar kind of diagnostic yield, having 12 novel conclusive diagnoses and 4 possible diagnoses, again leading to an overall increase of diagnostic yield of approximately 10 to 15%. Importantly to notice, this was not sequenced on the latest technology of the hi-fi sequencing opportunities that PacBio now offers, and it was also only sequenced at 10x. So this also shows you that there's a lot of low-hanging fruit to detect for these kind of cases, not only looking at 30x genomes. One of the examples of these studies, this is 2 cases with singleton males with neuromuscular disorders in the clinical diagnosis of neuromuscular dystrophies, where they expected DMD to be involved. And this is the first patient where DMD is breaking in an inversion through these 2 genes, and in the reverse orientating, disrupting this DMD gene. But on this, in the second patient, you can see that the breakpoint is slightly further ahead for the same type of rearrangement, so also an inversion. And this is also breaking the NHS gene. And interestingly enough, DMD is a known disease gene for the muscular dystrophy, but this other gene, NHS, is known for Nance-Horan syndrome. And retrospective phenotyping of this case also shows that there's features belonging to these patients that are related to this syndrome. So also showing that sometimes the complexity of these kind of inversions also explain part of the phenotypic spectrum observed in these type of patients. Just another example, because we've been working on this syndrome for quite a while, and this is very exciting to us at the moment and only recently discovered. So this is a case where the clinical pediatric neurologist says this patient must have GLUT1 deficiency. She has all the clinical and biochemical hallmarks of this syndrome. Why does she not have a mutation in a related gene, which is SLC2A1? So using long-read genome sequencing, we stumbled across this locus here on chromosome 1. And this was actually a translocation. If we look and follow these reads, the colors are again the haplotypes, but we have not been groups in haplotypes. You can see that those are not belonging to chromosome 1, but they in fact belong to chromosome 16. And if we zoom into this locus here on this breakpoint on chromosome 16, we actually notice that this is a locus where, lo and behold, this SLC2A1 gene is located. So this is not, well, I would call this just coincidence, especially not given that this is a locus where previously a publication has been mentioning that this is an enhancer specifically for the GLUT1 region. And this means that if you break this here, at this locus here for the breakpoint where the translocation occurs, you actually disrupt the STAT domain structure. And I'm sure that you have looked at the webinar from Marta Spielman, who has probably shown you a lot more on how to interpret these regions. But if you, of course, take away the regulatory elements of this gene, you disrupt how this gene is expressed. And also this is what we believe is going on with this patient, explaining why, despite not having a coding variant in SLC2A1, the patient may still have GLUT1 deficiency by disrupting the organization of this locus. At this moment, we're still analyzing this cohort of 100 patients that was selectively included because of their critical phenotype. And so far, we have analyzed 51 families, and we are already at the diagnostic yield of approximately 15%. So we're again at par with all of the other studies that have been published so far on increasing diagnostic yields. So having now covered two of our most important goals, challenging long-read genome sequencing for difficult to detect variants and seeing whether it can actually increase diagnostic yields, we moved on to our next main study. And that is, if this is all true, can you then also use this as a generic first-tier test for rare disease diagnosis? And again, this requires a very slightly different setup in study design, because whereas first we have looked at replacing one test, if this was a very challenging one, we have looked at what if everyone is still negative, can you then increase diagnostic yield? By starting from scratch, where the patients get their first diagnosis, then do one long-read genome and to see whether you can replace all of the diagnostic testing while still increasing efficiency and diagnostic yield, is something that had not been done before. So this is why we started this study in this design. To give you slightly more details on the study design, this means that we have a genetic referral, and in parallel to standard of care, but in a retrospective manner, we now sequenced in total 1,000 samples from the year 2022 that are representative for our diagnostic lab in the number of diagnostic referrals for this clinical indication, the diagnostic yield obtained, and the use of all the various standard of care workflows. And as I said, this is a retrospective, prospective parallel design because we started blinded to what was the outcome of standard of care by dedicated team and workflow, and no one knew the outcome before actually both tracks were finished. And then at the back end, we compared the outcomes to see what was the differences. Slightly more details on the cohort. So the 1,000 samples represented 832 different index patients that in standard of care were tested by a combination of genome-wide approaches, either arrays, carrier typing, and exome sequencing, or targeted analysis, here displayed in a different plot. If we look at all of our samples, approximately 750 were sequenced as a singleton at 30x, and for 84 we did trio-based analysis, which is in line with our average distribution of how many samples get sequencing as trios versus how many are done as a singleton. All of the samples were processed between August 24 and January 25, showing you that you can sequence relatively easily 1,000 samples in approximately 4 or 5 months. And that also helped us to understand how feasible it will be to sequence large scale should the results of this study be positive. Again, our bioinformatic and automation of the library prep processes. So to give you more insights, we by the time moved to the Revio systems. We use the Revio version 1 chemistry, and we have a relatively semi-automated bioinformatics pipeline that takes data to variant annotated files in approximately 16 hours. We sequence 48 to 72 samples a week to reach the 1,000 samples in the time span just mentioned, and we have 3 to 4 Revio systems that are availability to get this done. We have a very nice bioinformatics team who provided us with the blinded team the graphical user interface for variant filtering and prioritization. This allowed us to mimic the exact variant interpretation as was done in routine care by looking for the right gene panels, the different tabs here on top, listing the different variant types or the different tools needed for the variant identification. And using here, for instance, you see here an example of the CHEK2 variant. We get all of the information of relevance for interpretation right at our doorstep by just clicking some of the routine filters used in our in-house database. Now coming to the direct comparison of standard of care to hi-fi genomes. And what I'm going to show you here is our diagnostic yield using the standard of care in the 832 different index cases. And this is data obtained from the hi-fi genomes. So what you can see right away that the diagnostic yield in standard of care in this cohort was 15% and 18% in hi-fi genomes. So quickly said, there is an increase of diagnostic yield by 3%. The more important question of this study is, however, not whether you can increase the diagnostic yield, but whether the diagnoses that you get in standard of care are the same ones as that you can get using the long-read genomes. Because if this is 18% of different patients, then these 15%, we're still having some kind of a problem because, of course, you do not want to miss that 15% that you would pick up in standard of care. So here in the 3x3 table, I'm going to show you the direct comparisons of these different outcomes. With here in green, the conclusive diagnosis in standard of care and the conclusive diagnosis in hi-fi genomes, the possible diagnosis in standard of care and the possible diagnosis in hi-fi genomes, and the negative, so no conclusive diagnosis identified in either of the technologies. And this is the concordance for these samples. Of course, the ones that are still left empty are the ones where differences occur. If we first look at the standard of care positive cases, so there is a conclusive diagnosis obtained, but it is negative in hi-fi genomes, and we looked into the details on why that was, we see that out of the 832 cases, we missed one variant that was a low-level mosaic, and we missed one variant because of that sample having a relative low overall coverage. We found two more variants, or not two more variants, we interpreted variants in a different way than were initially identified in standard of care compared to the original diagnosis. And these were variants that by looking at the long-read genomes, we could actually phase. And the variants were, hopefully, at least that was the assumption because of the reporting, that they were located in trans to explain that they were both located two different haplotypes. But those did not explain the disease because they were in cis. More importantly, perhaps, is this 24. So these patients actually got a conclusive diagnosis from long-read genome sequencing, but a possible diagnosis from the standard of care. So this is enhancing the diagnostic yield. 21 of these 24 were because the variants could be identified as being in trans. One was a novel CNV, one was a novel small variant. And for one CNV that was already known, we could now tell where the variant was located and therefore also explain how this variant explains the disease. For two of the patients that were negative in standard of care, we now found two variants, both structural variants, that are either conclusively explaining the patient's phenotype or are still under further evaluation to see how this variant links to the disease. But it seems likely that this is also a conclusive diagnosis, one if we would move slightly beyond time today. So if we look at the first results of this study, we can see that 96% at least is in overall concordance. So this is relatively high. We only miss a few, and we can explain why we miss them. And we see a lot of improved diagnosis, mostly from better interpretation of variants that were already identified, but also the ones that we find new, again, have an overrepresentation of structural variation as what may have expected based on genome sequencing using long reads. Just showing some examples of what the new variants looked like that had not been identified before. So this is a case that is clinically suspected of Hopwood-Thompson syndrome, which is caused by recessive variants in this QL4 gene. This variant was already known, and we, of course, recover this in the long reads. Again, this is an IGV screenshot with the reads grouped by haplotype. And this is the second haplotype where we can now see that there's a small deletion in an intron. And that intron had not been covered in the standard of care experiments where whereas it is known that this gene, or at least it has been recently described, that smaller intronic deletions, given the small size of the introns, are so disturbing that the normal splicing can no longer take place. So we believe that this deletion may also explain the patient's phenotype in this case. This is another interesting example where the green arrows highlighting here where the increase in coverage occurs, and also, again, the so-called rainbow reads indicate that there's something going on. And in this case, this is a tandem duplication. This gene FBN1 is causal for a known syndrome in the cardio-Marfan spectrum. This original standard of care test was exome sequencing in this case, and the original outcome reported in standard of care was that there was a VUS reported, so a variant of unknown significance in the FBN2 gene, so a totally different gene, part of the same disease spectrum, but not fitting the patient's phenotypic resemblance. And we now see that in this gene, which is a better fit, there is a clear duplication orientated in tandem, intragenic, also disrupting this gene's function. And you can clearly see this is located on one of the haplotypes and not on both. So here we also have a new variant explaining the patient's disease. I'll go, for the sake of time, relatively quickly over this. But what we have also looked and noticed is that phasing is one of the main assets of having haplotyped information, and that this is at long reads far better than in short reads. So in short reads, you can average phase 2 variants apart of 800 kb, whereas if you have long reads, you can go up until almost 200 kb for 50% of them. So we looked at how well all of the known disease genes today are actually phaseable, assuming that you have 2 heterozygous mutations in the gene. And we can see that, of course, as you expect, that if your gene size increases, it becomes more difficult to fully phase an entire gene. But if you look at it from a different perspective of for how many genes can you actually say that you have seen 2 alleles, then you can see that's almost 96% of all of the disease gene panels known to date. Again, similar to before, the 5-methyl-C helping you in a methylation. Imprinting disorders you can detect, although you can clearly see that this is not a cause of disease in this case, and because of the nicely methylated and unmethylated region here. And this is an example, again, of one of the patients having a skewed X inactivation in the lower panel, whereas a control is shown on top. More importantly, and not shown so far, is that, of course, if you have this DNA methylation readout, you can also read out the methylation profiles. And for many of the genes involved in glomerulopathies, it is now known that they cause a so-called epigenetic signature. And you can also, of course, sort of resemble that in a long-read genome. So what you can see here are 3 cases, each of them having a gene, a variant in a gene that causes such a disease in which a episignature is known. And you can see that those patients clearly are the outliers when compared to the remainder of the samples in this series. Very last, now knowing that you can very well resemble standard of care and also reach some improvements, the question becomes relevant. It's nice that you can do this for 832 samples, but what if you would do this for all of the samples that you will see on a yearly basis? Having the input of this series, we therefore modeled our genome first approach using all of our data of 2024. And this is of interest because in 2022, we did not do so much of the short read genome as a first-year test in our standard of care. So if we would just randomly compare this outcome to our current diagnostic outcome, we may not have the best comparison, as short read genome sequencing was not being done in 2022. So in our model, we took all of the cases for which we had information available that it was a DNA-based test on isolated from EDTA for a patient looking for a genetic diagnosis in the indexes only. And for that, we then modeled all of the data that I've shown you so far. So we modeled whether the variant was detectable in a 30x genome. So taking into account that the repeat expansions are challenging, that low mosaicism is very difficult to do. And of course, mitochondrial DNA, not so much spend time on here today in the discussion, but of course, having the circular DNA makes it very difficult to sequence it in a long-read genome. We also took advantage of what we have learned on the feasibility, the chance of uncovering the second pathogenic variant in a recessive disease gene, and the possibility of using those 5MC readouts for the interpretation of variants of unknown significance related to episignatures. And of course, while we were doing this study, Many larger scale studies were published on the increased diagnostic yield by large scale case series. And for all of the negative cases, we extrapolated the numbers of our diagnostic requests in 2024 to what that would mean in reporting increased diagnostic yield of our 2024 data. This is a more difficult to read schematic plot, but if we go through this step by step, we have approximately 2,500 conclusive diagnoses in 2024, of which the vast majority is recapitulated using long-read genome sequencing on the right-hand side of this plot. But 128 will become negative should you do long-read genome sequencing. And this is how you can follow all of these arrows. If we zoom into these arrows, we can highlight again the same observations as we obtained generating the real data. So 170 patients will become negative because of the technological challenges associated with the hi-fi genome sequencing technology. 233 cases will become positive, irrespective of being negative or a possible diagnosis at the moment because of better interpretation. And for 288 patients, we expect to either uncover a second recessive variant or have an increased diagnostic yield because of uncovering a new variant that had not been identified so far. So if we put all of that together and we take our 2024 cohort as a whole, we believe that if we would have done a long-read genome sequencing first strategy, that the diagnostic yield would have been 2.5% higher than currently achieved in standard of care, without looking into, not even including all of the novel possible diagnoses that you would find by the novel, more difficult to challenging interpretation of variation that would require additional follow-up. But this is simply only looking at the genes that are currently analyzed in standard of care for the same patient in the same way, but only exchanging the underlying technology. So the summary would be that we believe that genome sequencing, long-read genome sequencing, can be implemented as an efficient workflow, that the increase of diagnostic yield may be expected specifically for the undiagnosed rare diseases through the identification of novel structural variants, and that if you would replace all standard of care, that you would actually have a very high concordance, but have the bonus of the overall wins that you get from better interpretation and variant detection. And that our model also suggests that if you, at least for our diagnostic lab, look at all of these data, that you will enhance the variant interpretation by phasing and methylation information, but also the detection of new variants that so far have remained undetected or undetectable from standard of care technologies. So that means that we believe that long-read genome sequencing can be used as a generic first-tier test, that it can replace the other test modalities for germline-based diagnostics at the moment already, and that we can actually move from the interpretation of variants in its isolation, but more to the context of its alleles and its haplotypes. And with that allows us to increase the diagnostic yield. And to put the proof of the pudding in eating it, we have actually started the implementation of long-read genome sequencing in October 2025 in our lab, and expect to do 5,000 long-read genomes this year, not so much for the interpretation of the entire genome sequence in this way, but merely to have the benefit of the haplotyping, the phasing, and the ease of interpretation, not having to do 5 different tests sometimes, but having it all in one go, ready for interpretation. And with that, I would like to acknowledge all the people involved and thank them for all of the hard work they've been doing, and especially now in the phase where we are wrapping up this manuscript. And thank you so much for your attention.
Lisenka, wow, just absolutely phenomenal. I know, and that last slide sort of truly reflects the work of a fantastic team that have really driven forward this work over the last couple of years. As you can imagine, there's lots of questions for you, so I'll try and pick out some, and they range across some sort of quite technical things to some that are more sort of philosophical about the approach. So let me just pick out a couple. So one question from Denny Pop is just asking about the titration curves around the coverage that you described. Do you have any evidence or think that if you extrapolated to 40 times or 60 times, that actually you would get any greater yield, or do you think that there's good evidence that 30-fold is going to be enough?
I don't think we've done that analysis, but it would be not too difficult to do, to just sequence 2 samples and just pull the data and look at it. But given our current estimates that for over 95% of all the relevant disease genes known today, so staying within that clinical relevant context, we see 2 haplotypes. I'm not too much concerned that we actually miss an awful lot of variation. Will you gain more? Sure, but I think we're almost at the plateau level, and the plateau is never a plateau, but there's always, you need to sequence a lot deeper in order to then gain like 0.1% or so additional variance. So I think we are with 30x definitely at the plateau, and the question I think is how low you would actually be able to go downwards, not to lose too much, rather than spend a whole lot more money than to find the additional one.
Yeah, and you sort of touched on that when you looked at 20-fold and below, and there's a bit of a significant drop off below that, and some of the questions do
the data that I did not show you is that we have done in the 2024 models, we have looked at what is the most contributing variant type, and that's of course still the SNVs, indels, and the CNVs. And the impact analysis of going lower in coverage on those variant types is severely less of an impact on the negative side than on, for instance, tandem repeat expansions. But then again, the vast majority of our tandem repeat expansions are the GA-based ones that we see in our hands, or in our practice, I should say. So this may be very different for other labs. So everyone should do a sort of similar kind of analysis. So then if you couldn't find it at 30x, you can also not find it at 10x. So for us, that would not necessarily lead to an awful lot of differences in the outcomes presented here.
Yeah. And kind of one of the questions relates to sort of some of the sample processing and DNA handling before you actually do the long read sequencing. Are you able to work with stored DNA samples? Do the new eDTA samples need to be processed in a particular way? And are all these samples extracted from blood, or have you tried saliva or other tissue types?
We have done all of the things that you just asked, but to keep the very long story short, all of the samples that I showed you of the 832 comparison data sets, as well as the hundreds that were very challenging to detect in long read or in short read genomes and now tested for long reads, were all banked archived samples, sometimes even up to 10 to 15 years. That means that you can, at least for the PacBio-based long reads, and I'm not sure how this works for ONT because we have not tested that, our methods of isolation are at least, and storing the DNA for a longer period of time, are sufficient to still generate 20x reads without any troubles.
Okay, that's good to hear. Now, as you can imagine, lots of questions about how much it all costs. It's very difficult because you've been in a relationship with the company and doing research, and so it's different. But have you got any ballpark sort of sense of where it is now? And of course, our expectation is that this will change a lot.
Yeah, so let's start with where we are today, because I think it's no use to look backward on how much we spent on it, because we were early adapters, and that also means that you pay a little more than you would do now. I think the announcements today are roughly between $300, $350 or so dollars predicted.
Okay.
That is almost at par with short read genome sequencing, depending on how you do your short read genome sequencing, of course. And what we aim to do is not necessarily generate an awful lot of data for everyone to interpret, but merely to exchange what we currently generate on data production. So if we would today do an exome and an MLPA, that is almost at the same cost as now doing one long read genome. So whereas we have not done all of the micro costing, because of course then you also need to take into account the reductions in costs because of phasing out of workflows, we already believe that in our hands we could almost get it to a cost neutral transition to long read genomes for all of the samples that we have. Having taken into account that we soon expect to do 2 samples on one flow cell, which is one of the announcements PacBio has made at ESHG and ASHG, where the cost, of course, would drastically reduce.
Yes, that would make a huge difference. And
Yeah.
through your answer, you've answered a number of people's questions. So, Lisenka, we don't have any more time for any questions. So, thank you so much for such a wonderful presentation. It's a wonderful way to finish this first year of ESHG webinars. In the new year, our first webinar will be in January, on the 28th of January, and it's at a different time. So please note it's at 1400 hours Central European time, and that's because Sarah Norris is going to be presenting and is joining us from Australia. And so the time difference, she will just be still awake if we are all able to listen to her and join her at 2 o'clock. So we're really excited to hear about her work on sequencing in the newborn project that they're doing in Australia. So I hope you all have a wonderful festive period and new year. And thank you for joining us. And thanks once again to Lisenka.